When Einstein learned of Heisenberg's theory of matrix mechanics, or how many of the rules in the macroscopic world don't apply in the subatomic world, he was deeply troubled. In fact, even though Albert Einstein won a Nobel Prize in his work with quantum mechanics, he was one of its greatest critics. Einstein actually insisted that God does not play dice with the universe, and therefore there is no room for randomness in physical theory. An example of this randomness occurs in our everyday lives. For example, if we examine common materials such as silver and gold at 150 nanometers, or about 1,000th the width of a human hair, the items have drastically different properties. This difference in properties, however difficult to understand, has proven to have beneficial applications in the healthcare field. Silver, harmless in jewelry, is deadly for human cells microscopically, and gold can be heated to over 200 degrees Celsius within a matter of seconds with just the rays of the sun. It is precisely this interaction between light and nanoparticles that scientists at the Niels Bohr Institute are studying to eradicate diseases like cancer. The size and shape of the nanoparticle play a crucial role in how the nanoparticle absorbs light. When light strikes a metallic nanoparticle, the electrons, which are in a loose cloud within the metal, begin to fluctuate in time with the incoming light. If the size of the frequency exactly matches the size of the nanoparticle, then the frequency is amplified. This is called resonance. Resonance is a phenomenon in physics in which a vibrating system or external force drives another system to vibrate with greater amplitude. Imagine pushing a child on a swing. When pushing, you need to push at the right time, with the right force, and with the right location. Otherwise, you might end up stopping the swing altogether. It is the same with the resonance of nanoparticles. When light is at just the right frequency, strikes the nanoparticle, a very large fraction of the energy is actually absorbed by the particle. The closer the light frequency is to the resonance of the particle, the more energy is absorbed by the particle. Heating a metallic nanoparticle that is at resonance becomes interesting because they share electrons that sit in a loose cloud in the middle. When light is sent through a nanoparticle, the electron cloud fluctuates with the light. And if the light exactly matched the resonance of the nanoparticle, then there will be a very strong amplification of the frequency, leading to a tremendous absorption of light. The absorbed energy then dissipates as heat into the surroundings, away from the surface of the nanoparticle. If a gold nanoparticle of 100 nanometers is heated with wavelengths that may not even be perfectly resonant with the nanoparticle, it can easily be heated up to 700 degrees Celsius. Nanoparticles can also be tracked in 3 dimensions, moved around, and controlled. So how can nanoparticles aid in the fight against cancer? A technology, abbreviated LANTERN, is called laser-activated nanoparticles for tumor elimination. A lot of the technology is explained in the title itself. Cancer cells absorb gold nanoparticles through a natural process called endocytosis. The tumor is then flashed with near-infrared light. Near-infrared laser light, also known as NIR, ranges anywhere between 750 nanometers and 950 nanometers. In fact, this is the same wavelength of light used in laser hair removal therapy. This wavelength of light can easily penetrate through tissue walls without causing burn damage. Even though the nanoparticle and the light that it's being shot at is not in resonance, the nanoparticle still gets heated. The heated nanoparticle then causes the cell to break down. So how hot can human cells get before they stop functioning? Well, cellular disruption causes cell death long before the entire contents of the cell die off, and cellular disruption occurs at the level where protein denaturization occurs. Protein denaturization is a process in which nucleic acids lose the structure present in their native state. For humans, protein denaturization varies from location to location. Most cells die around 40 degrees Celsius, 3 degrees Celsius over body's temperature. The enthalpy of reaction on minute scales may be much higher than this, but temperatures for the system in equilibria will not exceed standard human body temperature, as modified by circadian rhythms. This treatment has been tested on mice, and it has been demonstrated that the cancer tumors become considerably damaged. Nanoparticle therapy, whether it be through heat or drugs, is a much better treatment option compared to traditional radiation and chemotherapy, which have major side effects because the treatments cannot be localized. Furthermore, traditional cancer therapies do not eradicate CSCs or cancer stem cells. Cancer stem cells are cancer cells found within tumors or metastatic cancers that possess the characteristics of normal stem cells, including the heightened ability to give rise to cancer. Many people go through the treatment only to have the cancer come back again, and nanoparticle therapy ensures that this won't happen. Some of the next steps of this technology involve trying to get the technology to work when the tumor cells has metastasized and entered the bloodstream, or coating the nanoparticles with chemotherapy drugs that's released when the particles become heat activated.